Hello guys, my name is Dr. Varun and you are watching Chlorophyll classes only on YouTube. Well, this is for class 12th video and we're gonna be studying about the Operon. Till now what we have studied, we have covering the chapter molecular basis of inheritance, and today is the last day of this chapter where we will be covering three last topics. One is Operon, next is Human Genome Project, and the last one is DNA fingerprinting. This topic, try to understand from here, and every statement in the NCRT would be a small sweet piece of cake to walk over it. Okay, let's just start with it. What is Operon? Operons are what? These are group of genes working together to do one function. Where it will occur now, and more over to that where we usually found it. We usually see this in the prokaryotes. Okay, usually seen in prokaryotic cells. Now what is that? If someone wants to tell that how we will explain an operon is what? These are the group of genes working together for a specific function. Examples in prokaryotes, there are many examples like lac operon, his operon, tryptophan operon, valine operon or well operon, etc. These are the different operons which are working in the prokaryotes. Now the major question is that why that is taking place, because in prokaryotes you know that the regulation can occur either at the transcription level or at the translation level. At the transcription level, if amira is found and further these things, what could happen? This is another way of sorting out. Like for example, if the operon, how the operon works, we can make any part of this working stop and hold the process will stop. How that will take place? We're going to be taking an example of lac operon in it. So lac operon, first of all, it is what? It is a negative operon, because it won't start until something is given. Means it is what? The process of formation of the products which is needed by the end product of the group of genes would be stopped. Okay, it won't be starting up. Okay, to start it, something else is needed. So this is what the negative verb. Of course it has a positive effect also, but that is not in our syllabus. Our syllabus says that it's a negative operon, so we will be keeping our mindset like that. So these are what? Group of genes. Where the genes are present, it must be present on what? On the DNA, right? So let's just make a DNA and the part of the segment which is working on this and what it is working, then I will explain you. Now here, these are different genes which I'm gonna make now. Okay, okay, so these are the different genes. Let's just name them also. Okay, so we have I, P O Z Y A. These are the genes. Now these genes has a different naming also. So what are they? This genes I is known as inhibitor gene. Inhibitor gene. This O is known as operator, which is present in between. There was other gene is also present. This Z is Beagle genes. This Y is permease gene, and this A is transacetylase gene. Now the next thing that you have to remember is what this lac operon doing. This lac operon is present in prokaryotes. Let's take an example like E. coli. In case of E. coli first preferred food source is what? Glucose. But if glucose is not present, then what is present? Imagine that lactose is present. It has to digest lactose also to obtain the energy. So E. coli digest lactose through this operon. Now in this operon, what it does, this operon digest lactose into monosaccharide. What is the monosaccharides? Two monosaccharides are being formed by the lactose. Means lactose is made up of two monosaccharides. That is glucose plus galactose. So it breaks down into it and then it can eat it. It can obtain its energy from both of these products, because these are what? Monomers only. What is that? Biomolecules. We will learn it. Okay, that will also come in the book. Now just see that these genes, how they are working and helping this process. So first we're going to be seeing that since it is a negative operon, means it is not working. Okay, what does that means? Means in absence of lactose, what will happen? Let's see. Now see this, what gonna happen now. Now, and when nothing is happening, this inhibitor, okay, this inhibitor, since this is a gene, the gene will, if it has to do any work, it will only able to do when it will make the RNA first and the proteins will do the work. So this will make what? It will make a repressor RNA. This repressor mRNA. This repressor mRNA will translate and it will form what? A repressor protein. As this repressor protein will be formed, this protein, imagine it, this is it. Okay. So this protein will go and attached to this. It will attaches to operator, and that will do what? It will bind with it and hide and mask its activity and mask its activity. So since operator activity would be what? Operator will also make a RNA and the protein and the process of the next genes will be produced. But it will stop it. So what will happen? This binding will stop the operators from doing its activity. Means what will happen? These genes which are present, these genes are called as structural genes, because these are only going to be doing the major work. structural genes won't be able to form enzyme for digestion. So means understood that it is not able to do its work. Okay, so what was the thing? If you see that first point, second point, third point and fourth point, simple as that. So negatively it is working at the normal time when there is no lactose. But what if the lactose is present? Okay, so let's go to the next page. So next is in presence of lactose. So what will happen? Let's see. Let's just copy paste it. Now let's see what's gonna happen. This time, what happened? There will be what? Lactose is present. So I'm writing lactose is present here. Everywhere lactose is present. Now what this lactose will do, that all the time inhibitor will be doing what? Inhibitor will be making the protein. So inhibitor will be doing what? It will making the repressor mRNA. That repressor mRNA will be making the repressor protein. That is what? Like this. But this time this repressor protein is not able to do the work because of the lactose. So this is what? Lactose is present. This lactose will go and bind with it. Okay, so what happened finally? The lactose will bind with the repressor protein, and as it will bind, this will not be able to, okay, protein isn't able to mask operator gene. So operator gene, it's not able to go. Normally what it is doing, it is going and working with it, but it won't be able to go over here. So operator is free. Okay, so operator will say what? That operator will say that make enzymes. So means it will initiate the process. So now Z will make what? Beta galactosidase enzyme, which will do what? It will digest the lactose into monomeric unit, into monomers, respective monomers, which can be used up to get it. This Y will permease, will release what? Permease enzyme. This will do what? It increases the permeability of, cell towards lactose. Means more lactose will be coming inside the cell from the outside medium where it is present, and this transacetylase will help in the transfer process. So it will release the, so what will happen? The lactose will go and attaches to this. It will stop this repressor protein to go here. So this will start its work. What it will do? It will say that ZYA makes your enzymes. So they make the respectively RNA, and that will make the protein. So these are the final protein things that will be made it up, and they will do their work and leads to what? They will start digesting this lactose. Until when? Until this lactose which has binded to it is also being eaten up. And then what will happen? Enough amount of lactose is not left out, which can stop the repressor protein. So repressor proteins go and join with it, ultimately stopping this whole process. So this is what you have to remember. So now very important thing is that now little bit amount of lactose should always be present. Why? Because it will keeps the permeability open. Because if nothing is present, then from higher concentration means outside environment, it won't be coming inside the cell of the prokaryote. Understood? So this is a very important cell or solute solvent concept when the permeable membrane is present between them. Okay, so little bit amount of lactose should always be present. For lactose to keep it permeable. With this, your lac operon is over. There are other operons also present which is not in your syllabus, but they also works on the similar, either the positively or negative kind of way. Here the lactose is considered to be the inducer. Okay, so lactose would be what? Inducer, to induce the process for the operator to make the gene respectively of ZYA. Moving on to the next thing, after this comes our second topic of the day, that is Human Genome Project. What it is. Since we know that we are humans, me, you, everyone you see that looks somewhat different and has somewhat different capabilities, means there must be something different in me and you. To find out this, more questions like that would be what? To find out that what is the sequence of genes on our chromosome or on the DNA. We did what? We did Human Genome Project, where we took whole of our genome, means our genetic material, and try to dissect that what it does. So this was the mindset behind this whole project, and it is considered as one of the biggest project. Let's try to do and deal with most of the topic of it. So Human Genome Project, which is also called as HGP project. Okay, it was used or it was made to determine the genetic information of a given organism. Just humans? No, except us also certain organisms were researched on. Okay, moving on, this genetic, since the basic thing I have told you, this was launched in the year 1990s, and it's a very big project completed in 2001. Okay, so it's a 13 years long ambitious project. So it is considered as a mega project. In this first thing, with respect to human genome, so human genome has 3 into 10 raised to power 9 base pair. Okay, that is, genome means what? Haploid. If you remember in the initial, means this is what? Haploid content they are talking about. Diploid will be multiplied by 2. Okay, and now what is happening to understand that how mega this project is? If for the one base pair they are spending $3, it will cost what? It will cost $9 billion US. Okay, the estimated cost of the project. So this is the important pointers, you have to remember it like that. Further, if you want to type all the DNA sequences and each page has 1000 alphabets of the nucleotides, then 1000 page books, almost how many such? 3000 such books with 1000 pages, each page containing 1000 nucleotide bases written one after another would be needed. So this is another pointers over there. 1000 letters, 1000 pages, 3300 books, such books will be there. And to comprehend this much of information, we need what? New branches of the studies to store the information, and came what? There's the birth of bioinformatics, because to how to store the information of this scale. Okay, then so what is the goals of SGP? So it has what? To identify all the approximately genes which are present over there, 20,000 to 25,000 genes of the human DNA, to determine the 3 billion chemical base pairs which makes the human DNA, that which will be present, to store the information in the database. For that we have even developed bioinformatics. Then to improve the tools for the data analysis, transfer the related technology to other sector like industries where it could be helpful to them, and address the ethical, legal and social issue, which is ELSE with this. So goals of HGP is this. Now this project is a 13-year long project, and this project was done by multiple partners. So your name has to remember. First is the US Department. So first is the US Department of Energy and National Institute of Health. Both are from US. Okay, then this project was later on joined by, like earlier it was with the Wellcome Trust, second people, this is third. Okay, then there will be other partners like Japan, France, Germany, China and other later on contributed to its growth. Finally, it is completed in 2003, although not completely completed, but X and Y was still left. X was completed in 2023 itself. Okay, so it is this long. Still, we have to code for the Y. Okay, knowing that how much amount it is there, how much help it could do, we are doing it, and the other organisms were also being done. Like for example, those names are very important. So we have done for the bacteria, yeast, Canorohabititis elegans. This is what it is, a nematode, and did not cause any pathogen. So nematode, another example, means Schelminthes are two more examples, except the three examples given in your 11th class Schelminthes topic. Two are here in this biology also. One we are studying over here, elegans, and another one we're going to be studying in the biotech. Okay, then Drosophila, which is a fruit fly, plant like rice and Arabidopsis etc. were also sequenced. So this is an important topic. Now there are two different methodologies they took. One is that they take the DNA and one after one they keep on writing the sequences. That is called as sequence annotation. Another one is that they will express the small part of it and then they will write it that this will do this work. So these are the true way: expressed sequenced tag, small part they are expressing it up, or sequence annotation. Now how they are expressing that part? They are using the bacteria culture or the yeast culture. Okay, so commonly use the host are the bacteria and yeast, and over there they are, this is they that been called as, okay, for this process. Okay, we will learn this whole process and everything much about in the biotech. Okay, coming up to the next things, these fragments, how they are being used up and to form this process has been made by what? By the Friedrich Sanger, whose photo is also there in the our what we say thumbnail of the video as well. Now once we have done this sequences thing, the next thing is that this we have like this, you have see that human genome project representation, that this person's cell, then the DNA chromosome we have taken out, chromosomes we have unfolded, and whatever the information we put it into the this computer, that is bioinformatics. So everything has been tried to sum it over here. Now very important fact that I have told you beforehand is that the sequence of chromosome number 1 itself completed in 2006, but we said that most of the work is done is 2003, just the comprehending work was left, and still there are what 22 chromosome X and Y to yet to be completed. So they are not completed, but X is completed in 2023, which I have told you, completed in 2023. Another major challenge is that for the genetic is the physical map of it, to how to put it. Okay, so they have done what? They found that there are certain sites which are repeating multiple times and called as polymorphism. Okay, so polymorphism of the nucleotides help in doing it and making the small copies or the microsatellites which help in sequencing and later on DNA fingerprinting. So what are the science feature of human genome project? Means what we get at the end. So the goals are different and end results are different. Okay, so this is also called as outcomes. So for the board, this could be very important. So what are the outcomes? That this number of the nucleotide bases are present. Okay, then average there in an average gene there are 30,000 present. Biggest is present on the chromosome number 1 dystrophin, and dystrophin is the largest one containing 2.4 million genes, and the smallest is present on the Y chromosome. Then total number of estimated is 30,000, which is much lesser than what was earlier think, that is 80,000 or 1,40,000 genes. So means it's turns out that most of them are similar in all of them. 99.9% is similar, only 0.2% the difference that is coming up, making the all the difference things. So less than 50% of discovered genes have any function. We don't know any other functions what they are doing. Less than 2% code for the protein, means which whole of our body making up and doing the functions. Repeated sequences coming in the large portion of the genome, so we call it what polymorphism. Okay, and then repeated sequence are the stretch of DNA that are stretched many times and comes out 1,000 times, and they shed on light on that how the chromosome structure, dynamics and things are there. Okay, then chromosome number has the most genes 2968 and Y has the least, that is 231. Then there are what scientists identify that 1.4 million such locations are present where there is only single DNA nucleotide differences there, and that is known as single nucleotide polymorphism in one person to another and called SNPs. So between me and you, we have what SNPs present. Okay, and this information promised that how the chromosomal location, disease and all those things can be help us out. Then application and the future challenges, if you see that we use that in the DNA sequencing, tumor knowing, disease causing things, and where we can actually work on those things in repressing the things or maybe working it further with. So this is what its challenges and all those things. Then comes the DNA fingerprinting. DNA fingerprinting is what it is, that in 99.99% cases my DNA will be similar to yours, but the 0.1% that is present, which is approximately being what 3 into 10 raised to power 6 base pair, would be different between me, you and everyone, and that becomes a lot of possibilities. And that is what why my DNA will give us specific sequences if I will try to find it out. So this is what we're going to be seeing with the help of the repeated DNA and the satellite DNA. These satellite DNA, depending upon what they are rich and not rich, they comes up as a different types in me and you, and these things are known as what this pointer is known as polymorphism, means many different forms of this, and this polymorphism will be do what it is inheritable also. Okay, which is present in me, it will goes to my child also. Of course it will do the variations, means that my wife and what will be exposed and seen would be the case. Okay, now the next thing is that there are in DNA fingerprinting which is being formed by Alex Jeffrey. This person did what? This person found out that there are variable number tandem repeat. Certain things are repeating again and again and again on the DNA, and that is making what my DNA, your DNA different, as well as my DNA, my parents' DNA is similar. So how they do it? If you know the biotech, you will be able to understand. If not, then once we will do the biotech, we will come back on this topic. So don't worry that we are just here giving a quick brief of it. First of all, we will do what? Isolation of DNA. Then we digest the DNA with the restriction enzyme so that DNA can be smaller fragments. Then we put it on the electrophoresis where it will be separated out. Okay, after separating out, we put it in the nylon sheet, okay, or the nitrocellulose sheet. Okay, and then we will keep the hybrid, like divide it, split the DNA into half part and keep its probe. Probe means that something which has what? Radioactivity. We will understand in the biotech, don't worry over here. It will comes and join with it, because you know that if the single DNA is present, its opposite will pair with it. So if the opposite thing is available, we will pair it up, and this thing would be, then we will see that in the radiographs. So basically what we are doing, there are certain facts also, of course. So we are doing what? We are taking the DNA sample from the crime scene. So remember it like what? That there is what? A crime had taken place, and imagine that there is a person, like for example, a child named Nitish has been died. Now we will try to find and get the justice to this. Now when we are checking that as a detective, we found what? We found that there is a hair sample of other than Nitish present over there. So we did what? We took the hair sample. The hair is made up of what? Cells. So the cell will do what? The cell will have the DNA present. We took that DNA, we increase its number, and we put the radioisotope, half of it. Simultaneously, we took the suspect DNA, for example, A, B, and C people. For example, A, B and C people, we took it, we did whole process, we extracted their DNA, we divide their DNA. Once we divide their DNA, only the single strand is present. Now we have already bought suspected DNA. We will mix the suspected DNA with the A, B and C, and the suspected DNA will go and what attach to its complementary pair. Okay, because what happened, if it has ATC, it will have what T A G. Okay, it will go and attach like this, right? Whereas here it won't be able to attach. Where at those positions where the differences are coming up. Okay, it doesn't mean that all the places where my and your DNA is similar, 99.9%, it won't join. The other places it will go and join to that small parts which we have made it, and that's how we are able to find that who is the criminal over here. If you didn't understand, don't worry, I'm coming up with a very nice reel on it with an explanation of it. There you can learn it much better. I hope you have liked this video. I know that this is a kind of quick video compared to summarizing up, but we are coming up good questions and everything of this video. Plus we are coming up with a elaborated and good lectures classes for you on completion of board as well as NEET examination 2026. If you are in your 12th class currently and struggling that how you will complete your 11th class, then you can take this class and it will be like smooth like butter, where you are able to get 360 out of 360. And if there is a big announcement also there that if you take this class and you are able to get 360 out of 360 in your NEET 2026, then you will get absolutely this course free, means all your fees will be refunded on the day of result itself, making a win-win situation for you, and nothing like a scheme or course available in the market right now. If you are not preparing for NEET 2026, if you are preparing for CUETs or for the board examination only, then you can take the other course also, which is available at the similar cost like your neighborhood. For more details, you can check out on our Instagram handle, that is chlorophyll classes on the Instagram, and DM us and ask your queries. We are available all the time for you to help you out in that. Well, that's all for this video. If you like this video, give it a like, share it with your friends, and let them know that if they are struggling in biology, that could be done with me, by me. That's all for this video. Thank you.